All right. Good noon to you all. Hello. How are you guys? Hello, hello. Anybody here yet? Hey, hey. How you guys doing? I have some good news. I found the water filtration, the Lifesaver bottle that I was trying to tell you about the other day. Let me see what I'm gonna do is show you here. So I've got this Lifesaver bottle. It's kind of generically looks like a water bottle, but then what you can do is with the bottom, twist, pull it out, and then it pops. So essentially, this bottle is going to pump water that collects kind of on the outside here. You can kind of see inside there's these little grate things. There's a membrane inside there, and so if you fill the water bottle up, let me show you. If we fill this water bottle up, you can kind of see inside there's a place where cleaned water can fill, but we can also fill it from the other end. Let me put this back on. And essentially, we can get water in it from one side, and then, so we would fill the water bottle here, and that would essentially collect everywhere except for in the cleaned chamber. So that's kind of the outer side of that's the feed side is in that chamber, and then the permeate will come through where you're going to drink the water. So the permeate ends up in here, and the pressurization system is driven by hand using this hand pump. So when you pump it up, it applies pressure across that membrane, pushes the water through, and then of course you can directly drink it from the nozzle, as you would any water bottle. So that's pretty cool. This is an ultrafiltration membrane. I believe it's 0.22 microns. And so I kind of wanted to show you guys that. These are, you can buy them in a store. This one's a few years old, but called Lifesaver bottles. Kind of shows some more details of the mechanics here. No, this cannot make salt water drinkable. That's a good question, but this is doing ultrafiltration, the UF there. So salt will make its way through the membrane without any issue. So you're gonna, if you do it to salty water, you're gonna remove the particles and bacteria and most of the viruses from the salt water, but the salt itself will go through. I have another cool toy to show you, but it's more related to ultraviolet light. So I'll show you that in a little bit. Excuse me. When we get there. Okay. So today we're going over disinfection of water with mostly chemical, but then we're going to transition to UV. And your comment there, I often wonder why we can't make salt water drinkable in 2021. Well, we can. We absolutely can. We can do it in a variety of ways, but it's a matter of cost. When we talked about membranes, I think I discussed a little bit reverse osmosis. And if you take a really, really small pore size membrane and you drive a very large pressure across it, you can get the reverse of osmosis to happen, and that makes it so that you're removing the water from the salt. You're pushing the water away from the salt with that pressure gradient, and you can get clean water that way, but it takes a lot of energy. And likewise, and one of you emailed me about this, you can also have, you can distill the water or boil the water and collect the evaporation, but that takes a lot of heat energy to do it, right? So it's more about the economics of it, and I don't care how strong you are, you're not going to be able to hand pump a reverse osmosis membrane and get any meaningful amount of water through. Maybe you can make a fancy device and be pumping for hours and hours to get a water bottle that's filled with drinkable water from seawater, but it's just going to take a very, very long time and a lot of pressure exerted. So it's going to be a pretty hefty engineering challenge to make something manageable by hand with a meaningful amount of water. Okay, so that's kind of fun. Wanted to show you that. Last time we left off talking about this example problem here, where we have HOCl added to water and it dissociates, and there's a question about what fraction of it is in the HOCl form versus OCl minus. And so I wanted to come back to this problem and talk a little bit about the approach, because I was a little bit convoluted last time. So we're just briefly going to touch on this and then move on to the rest of our topic for today. There we go. Okay, so we have a certain amount of HOCl added. It's added as HOCl, 15 milligrams per liter of it, and then we know that the pH is 7, and it will dissociate accordingly into some portion of HOCl and some portion of OCl minus. So in this case, what the question is asking is percent HOCl at pH 7. If you recognize what this question is really asking for, it simplifies the process you need to do. A lot of times you will absolutely have to convert your mass into molar units, compare the molar units, and then give your final answer as maybe in molar, or maybe you have to convert it back to mass. In this case, we're just asking about the percentage, and we don't need a lot of additional parameters. One thing I did say was, let's assume that the Ka at 25 degrees Celsius for this was 10 to the negative 7.54. Okay, so at pH 7, we're looking for the percent of HOCl. Now there's a couple ways we could do that. We could look at the fraction of HOCl divided by the total amount of chlorine, which is simply HOCl plus OCl minus. Multiply this by 100%. That'll give us our answer. Or we could consider that we can simplify that, or think about it intuitively in a different manner, and simply say, well, if we have a fraction of, what fraction of the solution or of the chlorine is going to be in one form or the other, then we can know the percent of the total. So if we had 0.3, the fraction of HOCl is 0.3 compared to OCl minus, we can get a percentage answer from that way as well. So we can take that approach, or we can say some ratio. We'll say HOCl per OCl minus. And we can take that and say, let's say this is 1 to 2. That means that for every 1 HOCl, there's 2 OCl minus, which means we could treat that as 3 total and work from there. So we can get this fraction and say, okay, let's say if we take this is x, then we know that there are x number of HOCl per 1 OCl minus. So it's just rewriting it in a very simple term, right? That's something we can take a look at. And so then we could say HOCl, let's see, we can say that the percentage is going to be equal to x divided by 1 plus x. And that gets us into a form that looks a lot like this one if we simplify it. I think I'm doing that correct, right? So the amount that ratio here, HOCl divided by OCl minus, if you want the percentage from that, we have, let's see, did I do that right? I think it'll be easier if I actually work this out without symbols and just come back to the symbols in a moment once we have an example in front of us. So let's imagine, for an example, x equals 3. So that means HOCl equals 3 times the amount of OCl minus, right? So if that's our example, we can see that if x is equal to 3, then this equation becomes HOCl equals 3 times, so x times OCl minus, if we just flip it that way. From there, we can say, well, we have these in terms of one another. And so essentially what we're doing is we're then putting it into this equation, but you can kind of do it intuitively to get there. The other thing you could do is simply put in the numbers up front and solve for exactly the values, and you wouldn't be doing it quite symbolically like this, but you would be doing the exact same algebra. So whichever way you come to it, you're going to make use of the fact that HOCl divided by OCl minus equals this was our Ka over pH. And it doesn't really matter, and let me just double check that I got that the right order. So I'm sorry, H plus over Ka. It doesn't really matter what, which way you go about it, you're going to get the same result. You just, whichever way you think about it. So my point here is that if you're doing it intuitively, you can get there, and you will end up using essentially some form of this, maybe an algebraic simplification, or you can just directly use this concept in combination with the relationship here. And the reason I was saying I'm highlighting this ratio and what it means is because you can get the ratio very easily by this equation, which came from our base equation that said Ka equals the products times each other divided by the reactants. And maybe I'll say it this way. I'll say C to the C power times D to to the d power. Remember, this is multiplied here, divided by a to the a times b to the b. Okay, just pointing out that this guy, this equation gives us that formulation, and from that formulation we can get this type of information. It doesn't really matter how you get it there or what you do once you know this ratio. You yourself should be able to convert a ratio into a percentage. It doesn't have anything to do with chemistry. This is, if I tell you you have 4 green balls and 1 red ball, what percentage of those balls are green? You should be able to answer that. That's way beyond this class, right? That's way before. So I'm trying to get you to think outside of the scary chemistry bubble here and apply that knowledge that should be intuitive for you as an engineer and as somebody that's trained in math and problem solving. Okay. So the point was just being that we went from this equation. If there are any questions there, I'll try to clear it up a little further. So hopefully that helps. And you have this page as well from last time, a little more convoluted than I should have. Okay, so disinfection. We talked a bit about chlorine last time and the different chlorine forms and how we use it. We also talked about how we make use of how we're going to put that into an equation. I think we're going to elaborate on that a little more. Before we get there, I want to mention disinfection byproducts. Disinfection byproducts, or DBPs, are unwanted products of natural organic matter, which is whatever kind of organic molecules, large organic molecules that have who knows what structure, all sorts of plant material, soil material, organic stuff. If you think about why a bayou is tinted with a brown color, or why tea has a brown color, the tannins, the humic acids, the different things, these molecules will have small groups on them that can react with chlorine or bromine, iodine, fluorine, these halides. So most of the disinfection byproducts are halogenated organic compounds. And when we have these natural organic molecules, this natural organic matter, the molecules in them, we don't know exactly what's there. There's thousands of different ones, and what we're interested in is when we form these spots where we have some molecule, I'm saying R would be like all of this stuff, and then maybe we're interested in, on top of that, the one functional group on top of all of the other stuff. So if, from organic chemistry, this is kind of the way we talk about it. This would be R and then dash, and then we describe or write out whatever molecule we're interested in. So this thing being R, that's what I'm kind of referring to, is some molecule that's bigger, there's other stuff there. So we have, let's say, R, the molecule, and then on one end we have normally a methyl group. So that means we have a carbon with hydrogens. Now methane, by the way, is C with a hydrogen on every end. So a few different ways to write this, but this is methane, CH4. Okay? So something you're probably familiar with, that's a natural gas, essentially. So here we have R with a methyl group. So this is not a gas form, this is just connected to a bigger molecule. And what we're interested in particular are things like trihalomethanes. So when we use lots of chlorine, I'm just going to write Cl there. If we overchlorinate, then we could end up with R, and instead of hydrogens, we now have chlorine on each one of those. These tend to be more toxic. They can be carcinogenic, and we still have or excuse me, I wrote that wrong. We have and our methyl group here is now chlorinated. You may have heard chlorinated solvents. It's a similar concept here. These are harder to degrade, harder for biological entities to process and get rid of. So in high concentrations, these can be a problem. There are other classifications, haloacetic acids. So maybe instead of just the carbon here, there's the acetic acid group or vinegar, you've probably heard of. A vinegar type functional group there that has been chlorinated or brominated or whatever. So those are the types of things that we're looking at. And that's what we're concerned about in terms of over-chlorinating. Okay, so how do we know how much chlorine do we need? There's two reasons why we don't want to over-chlorinate. First is the disinfection byproducts. If we make too much of them, then we're going to have bad stuff in our water. We don't want to drink bad stuff. So that means we might want to try to remove some organic matter or treat it afterwards, after the disinfection, to remove those organic compounds that have the functional groups. The other reason would simply be we don't want excess cost. You know, we don't want to over-chlorinate our water because it's going to cost more to use that much chlorine. And maybe we also don't want too much chlorine in the water that we're distributing. And so we want to use the right amount, not too much more. It would be costly to remove it before we ship it. And it could harm our pipes if we send too much chlorine down the system. So we have a concept called contact time, or the CT concept. This is essentially a tool that can be used just like first order decay kinetics, except that it's incorporating the concentration of a disinfectant. So if you take a look at this rate equation, we're looking at how the rate at which pathogens N are being removed from water or destroyed, disinfected. It's as a function of time, so dN dt. And here we have negative K, C, N. Now, what you'll notice here is we have C to the N power and N to the 1 power. And that's a lowercase n here on the C. So a couple things to note here. We are looking at the change in N. So the change in N, that looks to be first order. But then there's a question about, well, is C changing? If C is changing, then that starts to look a lot like a second order equation because two of the variables are changing in here, especially if this N is 1. This formulation, excuse me, this formulation is called the Chick-Watson kinetics, and we call it pseudo first order. The reason we call it pseudo is because, as you can see, it might not always be first order. If C is constant, then it's first order. If C is varying, if the concentration of our disinfectant is changing over time, then we can't use first order kinetics because it's more complicated than that. So if N is our number concentration of pathogens, some number per liter or per milliliter, C then is our concentration of disinfectant in some units like milligrams per liter or millimoles per liter. This lowercase n is a dilution coefficient. And we are going to assume this little n right here is 1 for our class because we're assuming we have ideal reactors. If we had a non-ideal reactor, something like maybe my aquarium here, if I add some disinfectant right here, it's not necessarily going to be well mixed with the stuff underneath the gravel back there. So it's going to be that dilution is not going to happen perfectly. And so whatever I'm disinfecting here in this corner is not going to have necessarily the same amount of contact time with the water in other corners of the aquarium. So that's what this N is used for. And in our case, we're going to assume that we have ideal reactors, meaning that for plug flow reactors within the plug, it's well mixed, but not mixing otherwise. And for our batch and CSTR, they are perfectly mixed. So we're just going to leave that N as 1, although if you had to deal with a reactor that was not ideal, this might be a parameter that you adjust. We have K, which is the coefficient of specific lethality. This K here, this is going to be specific between your disinfectant, your chemical disinfectant, and the particular microorganism that you're looking at, that you're trying to disinfect. So K here, even though this is going to be a first order rate constant, it's going to have a more complicated set of units than usual because it has to also account for the concentration of our disinfectant. So typical units for our coefficient of specific lethality would be liters per milligram per minute or per time. So really what we're doing is the KCN, this has to go to mass, or well, really in this case, I guess number per time, right? Because that's our units right here. So that whole term has to go back to, at the end of the day, numbers per time. So that means K times, let's say, milligrams per liter times number per liter here ultimately has to end up as numbers per time. So here we have the K giving us liters per milligram per time. And I'm sorry, this should be milligrams. This is here, the N here, that's a concentration. So that's why this makes sense. Okay, so the N, it should have been number per liter. The number concentration per time is where these units go, because that's what these units are. Okay, given that, we see that the K has to have the liters per milligram in there to cancel out the constant here, the milligrams per liter, which is a constant amount of chlorine or ozone or whatever we're using as our disinfectant. Now, one thing we can do to simplify here is when C is constant, which we will assume, we can simplify and put a K star. So kind of like an effective rate constant. And then we can leave the K star as per minutes, because it's really going to incorporate, that K star is going to be K, the specific lethality, times the C to the N. So that's incorporating that concentration parameter there. And so this is then leaving us with just per minutes, like we've seen over and over again for a first order decay reaction. Okay, so it's all it is a very simple change here to incorporate the amount of chlorine or disinfectant. And really that CT concept is telling us that it doesn't matter if we have 1 milligram per liter of chlorine over 10 minutes or 1 minute with 10 milligrams per liter. So essentially the CT concept is saying, that 1 minute times 10 milligrams per liter is the same thing as 10 minutes times 1 milligram per liter. We'll get the same amount of disinfection based on either case, kinetic-wise. So it's relating, and it's saying what really matters is the amount of contact time you have between your disinfectant and your bacteria or virus or whatever you're disinfecting. Okay, so there's some data here kind of showing a case example from my research during grad school. And what I want to show here or highlight is there's a specific K for each pathogen to disinfectant pair. And when we study this, we'll take a look at how much disinfectant it required to achieve certain log removals of your pathogen. So in this case, we were inactivating MS2, which is a bacteriophage. A bacteriophage is a phage or a virus that infects bacteria. So these infect E. coli, and then we can visibly detect and see how many E. coli are missing or how many spots on a petri dish where we're growing E. coli, how many spots are there where MS2 is infecting it. So we can see MS2 dying really quickly given different concentrations of our disinfectant. And in this case, we were using a photochemical system to produce this reactive oxygen called singlet oxygen. So it's like a light-induced disinfection with some special materials. That was pretty cool, and we saw that when we were creating more of them or using stronger light, we had really rapid removal of our viruses. So we can use experimental studies, and we see, actually here's a good example. Here we have nonlinear kinetics. So this one was not pseudo first order, at least after the first little while. Maybe the first 10 minutes or something, maybe it was linear. We didn't really measure enough samples to tell, but we saw that eventually it kind of stopped working, probably because the amount of single oxygen that was being produced became limited by the materials getting all clogged up or something. So we can see then that depending on what type of disinfectant and the type of pathogen, they both are relevant. Okay, so here's an example problem, and I encourage you to grab a Excel sheet on your own and kind of work through this with me here. So this is example 10.4 from one of the textbooks online. It says the data for HOCl disinfection of poliovirus at a concentration of 1.8 milligrams per liter is shown below, adapted from a paper in 1979. The temperature was 20 degrees Celsius, the pH was 6, and it says determine the rate constant of inactivation assuming the Chick-Watson model applies with an N value of 1. Okay, so there's a couple things we could do here. It mentions HOCl disinfection at 1.8 milligrams per liter, and then it gives a temperature and a pH. So the temperature and pH really are talking about the Ka equals something at 20 degrees Celsius. It's temperature dependent, but only changes a little bit. So we have some sort of Ka. We could then deduce how much, our HOCl fraction is not actually the full 1.8 milligrams per liter. It's probably like 99% of that at pH 6. So we could go through all of that and do the same type of problem we started with at the beginning of this class. But what I want to highlight here is just using this raw data and assembling a graph and kind of observing what happens. So let's just assume that our HOCl is approximately 1.8 milligrams per liter. So we're just going to assume that it's all as HOCl. You know that that's not quite right, but we also, if we remember, the kind of break-even point where you have equal amounts of HOCl and OCl minus is at pH 7.5. And as we go lower, we get more and more HOCl. So maybe it's 1.75 or something, but we'll just go with 1.8 and keep it simple for this problem. Okay, so let's grab Excel. So we're going to want to put those numbers in here. And the answer, the question is, it says determine the rate constant assuming Chick-Watson kinetics. So we want K star as our answer. Actually, what we want first is, at the end of the day, is K equals. But along the way, we're going to probably find K star as that combination of the concentration of the chlorine times the coefficient of specific lethality. Okay? So let's say we have time in seconds and PFU per liter or whatever here. So 02468 6152 3000. Okay, so we have our data just like they put it there. And so we're going to insert a chart. I'm going to go ahead with a simple scatter chart, scatter plot. Don't need it that large. And so we see here we have kind of a curved line. And what that should remind you of is when we're taking a look at kinetics, and we talked about this earlier in the class, we actually would probably prefer having a linearized form of the kinetics. So this is essentially in a batch case. This is disinfection of a poliovirus. It doesn't give us anything about what reactor we're in. And so what I'll tell you right now is it's reasonable to assume this is a batch reactor because that's the simplest way to test kinetics. Almost always when we're dealing with kinetics, we try to avoid worrying about any mixing issues, any flow issues we'd have to calibrate for. It's just simpler to solve kinetic problems using a batch system. So we're going to just say this is a batch, which means, and we know it's Chick-Watson, so that's going to be first order decay. That means we can say V dN dt is equal to negative K C N. And so ultimately, if we convert this, we could say that's negative K star times N. And that's just like any other batch first order decay problem we've done, where we can say N over N naught is equal to e to the minus K star T. And finally, if we want this in linearized form, which we do want that for a for this kinetic analysis, we can say the natural log of N over N naught is equal to minus KT. In fact, if we do this in terms of just the raw number here, the number of platforming units in this, let's just say it's a 1 liter bottle and we don't even need the concentration. That would be fine. And by the way, I forgot the V here. Okay, so given the number in the volume, what we can say is that we have the natural log of N equals negative KT plus natural log of N naught. So that's either way we do it, we can do it in either form, and that's essentially our y equals mx plus b. Okay. I like to go ahead and keep it as this form so that we don't have to worry about the plus b, so it's just y equals mx. So what I'm going to do with the Excel sheet then is I'm going to convert it, do just a very easy operation. We're going to put this one aside. This one's the PFU versus time. Give me just a moment and I'll get there. On that. Sorry. Yeah, that should have been K star that I was writing. You're exactly right. Yeah, that should be K star because that's following straight from here. You can write it as K times C, but we'll use K star for the moment, and hopefully that'll make it clear. Thank you for catching that. Okay, so what I'm going to do in my Excel sheet is I'm going to set it up to linearize it like we have right here, the y equals mx. So I'm going to say natural log of n over n naught. That way we don't have to worry about the other portion, and we can say this is going to be equal to the natural log of the current number at the current time period divided by the number that we started with. And the number that we started with in the first point is exactly that same number. So that the natural log of 1 is 0. So that's correct. And we're going to fill this down. And by the way, a cool shortcut, if I didn't show you this already, if you're calling out some other cell and you want to put the dollar sign, you know, if you're familiar, you can put dollar sign before them. Those dollar signs freeze the pane, freeze the cell, so it can't, like if you copy down or whatever, it's going to keep the number. If you copy right, if you have the dollar sign before the letter, it's going to keep the letter. I learned this recently. If you press F4 while you're highlighting it, and I'm just hitting the F4 button, it will change through the different variations. It'll go dollar sign for both, dollar sign for the number, dollar sign for the letter, and then back to nothing. So pretty cool Excel highlight there in case you're interested. Okay, so now we have the natural log of n over n naught plotted as our y points. And so we're going to make a new chart. That's not what I wanted to do. We're going to select our data ourselves. This will be ln of n over n naught. Our x values are still time, and our y values here are this new natural log plot. Okay, so let me expand this a little bit. So here we have a much more linear form of the same plot. And now we can add a trend line and get the essentially a linear trend line where we have what I'm going to do is I'm going to set the intercept at 0 because we got rid of any, you know, the plus b and the y equals mx plus b. And I want the equation on the chart, and we might as well take a look at the r squared value. Okay, so now we have an equation, y equals negative 0.3758x. That's our y equals mx equation. In the r squared value, that's the variance, is looking pretty good. We want it to be close to 1. So we've effectively linearized it, and now we have this y. That y that we're observing is going to be the k star. So this is going to be negative 0.3758. That is in 1 over time, so that's per minute. No, per second, sorry. Because our x-axis was in seconds, right? Okay, so 0 seconds, 2, 4, 6, 8, 10 seconds. Yes, and I'll show that in just a second. Well, yeah, I'll go ahead and do that now. So the way I got the trend line, if I just have my data here, if you select the series here, I'm sure there's a few ways to do it, but what I did was I right-clicked on one of the data points, and I'm using Microsoft Excel, I think the 365 version, and on a PC. And so then I click Add Trend Line, and then I can select which option I want, and I set the intercept to 0, display the equation on the chart, and display the R squared value. So if that's something new for you, I would definitely encourage you to try it on your own, practice, familiarize yourself with it. Something that I would expect anyone dealing with data ought to know. Okay, so to get our final answer for the problem, to get K, which is essentially what we're looking for, right? Determine the rate constant of inactivation. Now, the question itself here is not great because it didn't really tell us if that's the coefficient of specific lethality or what. Oh, and no problem. Yeah, I totally get it if you haven't used it in a while. I'm not trying to say that you're wrong for not knowing it. Just seems like an important skill to me, so I'm just letting you all know that that's a good skill to have. And I don't mind answering questions like that either. Okay, so K then, if you remember, K is going to be equal to K times, it's the wrong pen. So K star is equal to K times C, right? So K then is equal to K star divided by C. And that's C to the n power, which is 1, right? So kind of just goes away. So then we can go back to our chart here and say K is equal to K star, and we're going to divide that by the 1.8 and get our final answer. This is in liters per milligram per second. And so that's how we would approach that problem. If we did the extra chemistry on the front end, maybe this would be, instead of 1.8, maybe it would be 1.75 or something, and that'll change a little bit. I'm just going to Control Z that and go make it come back. We'll just use this answer for writing up the problem here. So minus 2.09. Oops, 0.209, right? Yeah. Okay, so that's then a reasonable value for the disinfection rate constant, the coefficient of specific lethality for poliovirus. And the K star, while we're at it, was negative 0.376. Okay. Now, obviously, I'm not going to ask you to do a trend line analysis on an exam, but I did want to walk you through that, especially because you have to linearize it there, and that's kind of a good process to get a feel for how this data is applied in practice. Okay, so that's about it in terms of the chemical disinfection. It's relatively simple in terms of it's building exactly on what we've done before. The only difference is the K star and the K. So just keep that in mind. I think I've even put it on your equation sheet. I'll make sure to upload that or double check that I have uploaded that in a few minutes or at latest later today. So you'll have that to study with, but that's really the only difference. Otherwise, it's a first order decay reaction. I guess maybe I could tell you that, you know, no, it's always going to be first order decay. We're not even going to do anything different or weird. in reality, maybe there's a few exceptions. You could have something that's not quite first order or something that's closer to second order or something, but and if the disinfection chemical is not remaining constant, that would be a problem. But for our purposes, this is a first order decay. We might do it in a batch, we might do it in a CSTR, might do it in a plug flow reactor. We moved the exam to a week from today. So we have an exam review next class on Thursday. The homework will be due Thursday. We'll go over that together as an exam review, answer any questions you have, and then the exam will be a week from today. And again, my apologies about that. So next I want to go into ultraviolet disinfection. So here again I have a cool little toy for a portable water treatment device. I'll show you. So got a little, what's called a SteriPen. It is a device that we can take. It's battery powered, and it's good to disinfect a Nalgene bottle. And it essentially has a ultraviolet light here that is protected. It's in this case. It's designed so that it will only turn on if it's submerged in water, because we don't want to irradiate ourselves. It's a safety hazard. What I will say is, normally I would use it in an Nalgene bottle, but I should be able to do it in my glass of water here to demonstrate, so long as I'm not, this glass will absorb all of the UV rays. So I don't know if the battery is still good, but we'll check it out and we'll give it a try just in case. And so what I do is I submerge it, and then I press the button. There we go. And you can see we've got a light that's flashing, and then it says, it's kind of like a warning, and then it's going to disinfect, and you would stir it like that for some number of seconds. If I bring it out of the water, it automatically cuts off. So it automatically turns off when I brought it out of the water, and it'll stop the disinfection at that point. So now my water is extra clean. It did not go all the way through its cycle, and so it's telling me that, hey, I've got a red LED here saying that it did not finish. You need more time before it's safe. So that's a pretty cool device. I do believe there's going to be, you'd think so, the water that, my water would be the next level, but I don't normally add these extra treatment steps. Normally I just drink straight from the tap, or through my refrigerator water. But okay, so that's I could clean the fish's water with that, but I don't want to give it cancer or whatever. Probably harm the fish. I think they will eventually make LEDs that do that. We are starting to have LEDs that can emit in the UV range, but that was actually a fluorescent lamp, just a really small one, and a specialized plastic that would allow the rays to go, a lot of the UV to go out of that plastic. Okay, so ultraviolet disinfection. You kind of just had a quick picture there of what it would look like in reality. But I want to talk briefly about what is ultraviolet light and what part of it is useful. So we're pretty familiar with the electromagnetic spectrum, and we typically talk about it in terms of the wavelength. The length, that was a bad wave. So if we have a wave of light, we can take a look at the distance, the length, between two waves, and we would measure it between two peaks. This would be our lambda, or our wavelength. And depending on how much distance is between them, they could be a lot shorter, and just pretend those are all evenly spaced. If they're a lot shorter, that means more waves are hitting an object over time compared to these slower ones. Now, the light is traveling at the same speed, but there's a difference in how much energy is going, right? So even if they're both going at the speed of light, one is having, let's say, more photons hit the surface, the photons are hitting more frequently or with more energy, depending on how you look at it, compared to the two. And I think it's actually more energy. I don't think it's a difference in number of photons. But yeah, so it's a difference in energy. So the slower, not the slower, the longer wavelengths, the less energetic wavelengths are up here. So the longest visible light is around 780. That's getting to where we're starting to be in infrared. That'll be kind of a sort of a pinkish red type color. Whereas we go down and green, like a green laser pen is something like 532. And then we get into the blues and down to violet. Violet's around 400. That's about the limit of our visible perception. So the light you just saw me shine in my water bottle had two things. One is a blinking LED that was shining white to say, hey, it's active, it's about to start. And the other is that blue tint was actually some emission. That lamp was emitting strongly, something like here, and then a little bit, and a little bit like that. So what you were seeing was this portion. What we were not seeing was the ultraviolet light that was doing the disinfection over here. So in the case of our typical ultraviolet disinfection, we actually are using UVC. That's what we know as germicidal. And essentially the way it acts is DNA absorbs that wavelength and can cause problems with the DNA. UVB is more like what we think of when we think of a sunburn. It can literally cause a burn effect, damaging your cells based on a couple of different mechanisms. A little bit direct heat, but more so the energy of those photons hitting your skin can excite molecules, and those molecules, that excitation, there's a little bit of heat there for one. But the second part is that they can create these reactive radicals, a lot like those ones I showed you in the data that I collected here, which were disinfecting viruses. These reactive singlet oxygens, or radicals. If you've heard of free radicals or antioxidants, antioxidants are designed to capture those and quench them. The antioxidant is just that. And so the UVB there will create these radicals, and those radicals can then react with cellular components and create oxidative stress. And if you let that happen for long enough, or perhaps you're on some medication that acts as a sensitizer, it can really enhance that process and cause you to burn a lot worse. Those types of oxygen, those oxidative molecules, can cause cancer eventually, and other problems, but it's not as directly carcinogenic as damaging the DNA itself. So we could think of this as DNA here. We could think of UVB as sunburn, and UVA, and it's a spectrum, right? There's some overlap here, would be a suntan. If UVA is detrimental, does that mean infrared has some benefits? I wouldn't assume that's a corollary like that. The infrared, maybe there's some things that it's beneficial for. You can heat some stuff with it, but it doesn't always work that way, right? UVC is pretty damaging. X-rays are damaging if you put a lot of them, but can be really a non-factor with low doses. So it really kind of depends. Yeah, certainly infrared is not nearly as damaging. There might be some really vague concerns about something. I hear people concerned about it, but I haven't seen anything special. Yeah, you can certainly heat with it. I haven't seen firm evidence that infrared is damaging in some particular mechanistic way, but I do hear rumors that, oh, you shouldn't have too much infrared or whatever. I don't think it's a big problem. And it may even just be rumors. UVA, while there is some component that you can have those sunburns, UVA will trigger your body to create more antioxidants. So UVB will do the same, but it starts happening in the UVA. So you might start producing antioxidants. And if you've ever seen somebody that kind of cares about the way they look and are interested in having a nice suntan, really what they're doing is telling their body to make lots of antioxidants, because the pigment of your skin is essentially an antioxidant that is absorbing the light and reacting with the radicals to help protect the rest of your body. Now, obviously, some people take it too far, and you get the kind of a weathered type look after a while, after many years of doing it. But essentially, that is your body's defense mechanism. Yeah, and so that's the process that's going on. Your body's actually doing that on purpose, in some sense, in response to the exposure to ultraviolet light. So there may be some benefit to some UV. I am doubtful that the best thing is to always avoid all of the UV ever, because our bodies are designed with mechanisms in place to cope with it and to handle it. Of course, you can go overboard and have sunburns and melanoma and stuff like that. Also bad outcomes, but I don't know for sure that in fact, I've heard something about this that made sense to me as a photochemist, that some exposure can be a beneficial thing. So I have this example to show you of using UVC in a lab. I already showed you a light in person, so I think we can move on from that. Here's a couple designs that you might see in practice. A lot of times you'll have ultraviolet lights cased in these fancy quartz cylinders here that are kind of fragile, difficult to clean, and they get dirty, but you have to protect your actual fluorescent bulb, partly because the fluorescent has mercury in it, and you don't want mercury in your drinking water, obviously. So you have to protect them, and you have to protect them with something that does not block the light. And that's not a trivial thing. In ultraviolet range, almost everything absorbs the light. Oxygen in our atmosphere will absorb the light, so that the Steri pen that I was playing with earlier, if I did get the surface wet or something and I was shining it and looking at it, that would be a problem. Now, if I put it on the other side of my room and then turned it on that same way, unprotected, actually the oxygen and the ozone in the atmosphere, especially the oxygen because there's so much of it compared to ozone. Ozone absorbs really strongly though, which is why we like our ozone layer. By the time it reaches me, it'll pretty much be all the way gone if it's like 10 feet away or so, if it's not too strong of a light source. So it's kind of interesting. We can use that to our advantage, but we also have to be very careful. So I was telling you this glass here, I'm confident that it was absorbing all that UV because the glass itself is not a specialized quartz. It's just glass, like whatever glass they manufactured it with. And almost all glasses have silica oxide and some other things that oxide groups will be absorbing pretty much all of the light. I could have also worn, and I probably should have, worn some safety glasses, which I think I have some, and then had a second layer of protection there. So you can have it in that way, and I have also seen a kind of an advertisement type of thing where one company was making a sleeve or a pipe for the water that was transparent, and I think they did end up creating a polymer of some sort that did not attenuate too much of the UV, and then they put the UV tubes on the outside. So that really helped with the cleaning because you just clean this tube instead of the quartz sleeves around the bulbs themselves. So there's a few difficulties here and a few opportunities for ultraviolet emitting LEDs to start providing kind of new opportunities for disinfection applications. So thinking about the wavelength, I want to show you why the germicidal range works. So we're taking a look here, and the small UV light that I showed you at the beginning or earlier, that was emitting right here at this strong 254 nanometer peak. Oops, this is at 254 nanometers approximately. And you see it has almost nothing, and then it turns out it does have a little bit of something in the blue range over there, which is what you guys saw. There are also medium pressure mercury lamps. These are also fluorescent, just kind of pressurized differently. They require a little bit more infrastructure. They are not as efficient. You can see that the emissions peak looks like all this mess. This is the germicidal range. And we see that it doesn't have as much impact, but they do operate a little more energy efficiently, a lot more photons per energy. So there's kind of a trade-off, and sometimes large treatment plants might use this medium pressure lamps. But what I want to show you here is this curve right here. This is the germicidal effectiveness curve. That's essentially the absorption curve, the light absorption curve by DNA. And essentially, if DNA is absorbing these photons, it can cause the DNA to become damaged in certain spots, which I'll explain in a moment. And so this range is where ultraviolet light is going to be most effective. And you can imagine, since this is our typical mercury lamp, like I showed you, there is actually room to improve. If we could hit the peak here at maybe 265 or somewhere around there, we could potentially have better germicidal effect. And this is also why when we get up past 300 and around 300, like this here is kind of the UVB range. Wasn't necessarily very clear earlier. And then this would be the UVC range, all of that. And then we have UVA kind of up to the visible range. And so now you can kind of see why UVB has a little bit of germicidal effect, but mostly that's UVC is where the germicidal stuff is happening. So we are concerned about UVB for our skin in terms of cancer causing and the radicals that we might produce, whereas UVA, not as much, which is why we sometimes use a black light or if you've ever been like cosmic bowling or whatever, they'll have these black lights. Technically, it's maybe not the best thing to look at. Might be hard on your eyes, might not be great on your skin if it's a long-term exposure, but it's going to be similar to the UVA range that we experience under sunlight. Okay, so we can see it. There's some difference between different species here. This is showing MS2, which I talked about, and Cryptosporidium parvum. They have similar but slightly different DNA absorption spectra, essentially showing us that, yeah, there might be a little bit of difference between species, and that each type of organism is going to have a little bit different resistance or protection from UV, but ultimately the same principles are applying. Just one more confirmation of that. These are inactivation rate constants relative to the 254 mark. If they make the 254 mark, normalize that at 1, then they just check to see some organisms were more or less sensitive, but overall it was pretty much the same shape, right? If you go lower, which is not as practical usually, although maybe a couple of new lamp designs are coming out, fancy technologies like plasma stuff for like 220 might be pretty effective, but it's not going to go very far through the atmosphere. So all of these are similar because DNA is absorbing in a similar way. When we hit DNA with an ultraviolet light, it strikes these specific locations that have two thymines next to each other, and it causes them to polymerize or to dimerize, really, meaning that normally it was like, here's one, and here's one, and here's another, and these are acting like a zipper in DNA. These bases or base pairs where that code of the DNA fits together, and when the DNA needs to replicate, there's an enzyme that goes through and splits them all apart, and then comes back through later and joins them back together. But if you have this dimer here, essentially what happens is it's as if you took super glue and put it on one side of a zipper and froze those two teeth shut together, and then when you try to zip or unzip, it stops. It can't go past that point. You can't close this part or open that part. So it throws a wrench in the system, almost literally. And so if you have enough of these, then the organism won't be able to replicate properly, and you will have inactivated it. The organism might be alive still, but it can no longer replicate, so it has been sterilized, that particular one. Now, there are some organisms that can repair from DNA damage, especially if they're given some UVA or blue light. Some organisms are designed so that they have an enzyme specifically dedicated to repairing it that's driven by light. So it's either receiving lots of light, they have this process that goes to try to repair any DNA damage. It's not entirely effective. We can still destroy the DNA past the point where they can repair, but it is a small concern. So just a bit more of a technical look at it. When we have normal DNA, we have two T's in a row here. You'll see that's the place where we get the DNA dimerized, and that's where replication kind of stops when it reaches that point where it no longer knows what to do, where normally it would have gone through and replicated just fine. Okay, a couple things about ultraviolet light. When we consider applying ultraviolet light to disinfect water, we have to be concerned about the organic matter that might absorb the light. Like, if you add food coloring to water, it's going to absorb a particular color. Well, dissolved organic matter will do the same thing, except it absorbs a lot of UV light. So we have to be, concerned about how much organic matter is absorbing that light. We also have to be concerned about if particles are in the water to scatter the light, to reflect the light, or to refract the light. We see these ones here, and to potentially shade the bacteria. Maybe the bacteria is encapsulated inside a particle and protected, or it's just simply on the shaded side of a particle, maybe stuck to it, or just happens to be blocked. So we have to be somewhat concerned about particles in particular, no pun intended, interfering with the process of ultraviolet light. So we want clean water, and we don't want all the organic matter stuff. So just as an example, and this actually kind of showing this example will also tell you a little bit about the fundamental equation that drives disinfection and our understanding of light going through water. This is the Beard-Lambert law. You may have heard of it before. This is how light is absorbed. We have an initial light intensity and an transmitted light intensity through some distance of a cuvette or of some sample of water. And so we can make a relationship between these two that says the log of what we end with divided by what we start with is equal to a negative epsilon times the wavelength times the concentration of whatever is absorbing the light. So I mentioned that organic matter absorbs pretty strongly in the UV range. This is our important area, and we see this two different organic matter samples. This is Suwannee River organic matter, absorbs reasonably strongly, and then this other one, I don't remember the acronym there, USR, some other river, the natural organic matter from some other river absorbs up here. And that's pretty strong absorbance, especially if you compare that to nearly zero absorbance up in the visible range. So we can't really see the effects of this organic matter. Maybe it makes the water a little bit tan, but it's not doing much. Maybe a slight brown tint. Okay, so how do we put the UV light into a rate equation? Well, it's actually very simple. This is why I'm not spending much time on it. It's simply an equation that deals with the dN dt, and it has some relationship to a coefficient of specific lethality, just like our chemical, at some wavelength, the lambda, times the intensity at some wavelength, lambda. That just really means the dose. So this is the K and the C, so to speak. So if you remember, we had N, K, and C. That's essentially the same thing that's happening here. And that means that the dose, so the germicidal intensity would be the K times C, right? This is kind of like the K star. So I might give you a germicidal intensity, and that's your K, right? If I give you something that's in per time, that's essentially this K star, right? This is really all you need to work with. So I'm going to keep ultraviolet light problems simple because I always run out of time here because I like talking about UV. And at the end of the day, it really is simple. You dose it with this amount, it causes some amount of reaction, and you're killing your microorganisms. So I find for the ultraviolet stuff, it's quite interesting, but it's also quite simple in terms of how much dose did the bacteria receive or the virus receive, and it's really not adding much to anything we've learned aside from why it works, how it works, and that it's kind of cool that you can see it happen in action. So the dosage then is intensity times time. That correlates to that concept of the concentration times time in our CT concept, right? So both of these are effectively dose makes the poison type of systems where it's C times T, or in this case, intensity times time. Sometimes we would call this fluence, but it's just kind of another name for dosage. Okay, just elaborating a little bit here. We could say that the dosage is the average intensity times time, like I just said. Not a lot of important content here. Just kind of wanted to show you what that looks like if you were to be really explicit about it. You're not going to have a hard time on problems because it'll be pretty clear that it's this amount of dose or this K, and I may not even give you a UV question. We'll see. Okay, so that's all I've got for you. We'll have exam review on Thursday. Homeworks are due Thursday, and the exam 1 week from today. All right, so unless you have any questions for me real quick, have a good day, and I'll see you on Thursday. All right, bye for now. You too. I'm gonna feed my fish real quick because I need to anyway. So enjoy that. Yes, about the quizzes, they are still due Friday. I didn't see a reason to change those. We will have gone through all the content, and in some way, I think it's maybe better to get those out of the way before the exam. So thank you for the reminder. I meant to tell everyone. We have 3 quizzes due Friday. They are each 5 to 7 minutes or something. So make sure you do those. I'll try to send a reminder on Thursday and give a verbal reminder as well. All right, I'll see you guys on Thursday.